Welcome to class, y'all. Today we're going over the biochemical test of the Enterobacteriaceae, aka the 10 test tube test. So this is going to be a two-part series. Episode 1 of this is pretty much going to go over all the tests, positive and negatives, and pretty much just what to look for, the colors, and all that good stuff. Part 2 is going to be the identification and the big old chart that I had in the attached video. And this is the 10 test tube test that I'm talking about. So I'm going to start with left and go to right with each one of their names. Hello there. So first up we have the triple sugar iron agar slant, and then I'll go into detail about that. So there's basically no positive or negative per se for this test pretty much. You're just looking at color changing because there are 3 different sugars in there, and you're just testing to see if the bacteria can break down any of these sugars. So the red is an alkaline reaction, so it's becoming more basic. Normally happens closer to the slant versus the butt. The butt of the slant is down here. This is just the regular slant. And red, orange, pinky colors is an alkaline reaction. Yellow is an acidic reaction, which is very common to be found down in the butts because it's anaerobic down there. But you can have a few different reactions. You can have all acidic, which is A over A. You can have alkaline over acidic, which is K over A. You could also have gas production, so that's going to be A over A with the bottom A circled. Or you could have H2S production, which is any blackening in the tube, so that would be A over A. H2S positive is how you would denote that one. Alright, so next is the second tube, which is also the LIA slant, lysine iron agar. You're really just looking for any sort of red on the LDA. Everything else is negatives. The positive on the LDC does not matter. Indole test. Positive is red, negative is green. You're testing for an in-between molecule. I am speed running this because I have an exam to study for. Tube 4 is the methyl red. It's in the name. Positive is red. Any other color is a negative, even if it's brown. Tube 5 is Vogue's Scour, and positive is red, any other color is a negative. Tube 6 is the citrate slant. Neutral or negative is any sort of teal, positive is blue like that eraser. Tube 7, the OTC, not mistaken for the OCD, although that does end up on test tubes sometimes. Your positive is any color change and any growth, but it has to have the sterile mineral oil on the top because it's an anaerobic environment. If it doesn't have it, or there's no growth or color change, negative. My favorite test, the urea slant. We're testing for the presence of urease, an enzyme to break down urea. Negative or neutral is any shade of like salmon orange. I don't have that color. Positive is bright neon pink. I'll put a picture in later. Tube 9, the motility tube. A negative is no movement. You'll see a red line, and it might look a little fuzzy, but that's just from the stab into the agar. But if it's like cloudy and fuzzy, and then it looks like they've moved, it's positive. Lastly, you have the gelatin tube. Neutral or negative is no broken down gelatin, so the bacteria don't have the enzyme to break it down. Positive is a liquefied gelatin. No, do not base your results off of the tube directly out of the incubator because it will be melted. You don't want to test it like that. Let it sit at room temperature so you can see the difference between positive and negative. Yay, now you have the basics of the 10 test tube test. Huzzah! I'll post the image of all of what my tests were. So on the right is the TSI agar slant, and on the left is the gelatin slant, and it goes in the order as I described. And you see what I mean by neon pink for the urea slant? Yeah, that you want it Barbie pink.